Hello, everyone. If you came here to listen to some cool science, you came to the right place. Bless you. This is the Evenings at the Whitney seminar series. And my name is Jimmy Liao. I'm an associate professor of biology here. And like all the professors that work here, we are research faculty associated with the University of Florida on main campus. We just happen to be here in lovely St. Augustine, working full-time, where we train graduate students, teach classes over the summer, things like that. Before I introduce our speaker today, I just want to remind you, April 20th is our next Evenings at the Whitney. This is by Heather Mason-Jones, who's a professor of biology at the University of Tampa, and she's going to talk to us about the secrets of the Sweetings Pond seahorse. So that's kind of a cool, and Sweetings Pond is actually in, bless you, in Eleuthera, Bahamas. I know this because 2 years ago, I went snorkeling there specifically to see the seahorses, and they're actually pretty cool. Today's speaker is a distinct pleasure for me to introduce Sheila Patek. I've known Sheila probably, 20 years, maybe more. She works in the field of biomechanics, which is also my specialty. I work on fish. I'm the fish guy here. She's the shrimp, lobster, invertebrate person. Sheila started her scientific adventure as an undergrad at Harvard University. She then did a PhD at Duke University, and then moved to University of California, Berkeley on a prestigious Miller Fellowship. She was then so good that they kept her there as an assistant professor, and then she's actually been faculty at University of Massachusetts Amherst, and now is at Duke University. It was tough to distill Sheila's resume. It's quite impressive. She's had so many awards. To spit off a few, NSF Career, Guggenheim Fellowship, Radcliffe Fellowship at Harvard, Bartholomew Award at SICB. She was a Brilliant 10 scientist at Popular Science magazine. She gave a really cool TED Talk, which I actually watched again last night, which then trickled into more TED Talks, and I was up all night watching TED Talks, so thanks for that. But I saw that again. She does such cool research that she's sponsored by all sorts of diverse funding agencies, Department of Defense, National Geographic, National Science Foundation. Her work has been covered in all the popular media, New York Times, NPR, BBC Radio, History Channel, Weather Channel, Popular Mechanics, Wired Magazine. And what she does is she does many things. She's interested in sound production in lobsters. She studies the physics and physiology of fast. And you'll see that a little bit today in her talk. But more than that, she's a strong advocate for women in science, as well as the power of understanding basic science research, which is what we do at the Whitney Lab. She's an exemplary communicator for scientists, as well as politicians and the public alike. And so without further ado, it is my distinct pleasure to introduce Dr. Sheila Patek.
Wow, that was a really nice introduction. I think Jimmy made me nervous about myself. Anyway, it's such a pleasure to be here and such an honor to be introduced by an old friend and a very impressive scientist, as I'm sure many of you know. So I was preparing for this talk tonight, and my first plan was to regale you for a solid hour about really hardcore science, talking as fast as humanly possible. And then I was like, I'm kind of in this philosophical mood. And we're at an interesting point right now in the United States where it's hard not to be political, whichever side you're on, right? So I'm going to give a talk tonight about this world that I spend a lot of my effort in, which is understanding the outer limits of fast in biology, organisms that are doing things that engineers can't do, that show us new things that are possible. But I'm also going to talk about what is research, why do we bother, what does it mean, and more generally, what is the impact of this work that we do in biomechanics, that I do, that scientists do. So hopefully, I'm hoping, given the sort of many different places and thoughts where you all are coming from tonight, that I'll hit a few trigger points, maybe make some of you really happy, maybe make some of you feel a little irritated, we'll see, but hopefully engaged. That's my goal. So what I wanted to start with is just a simple question that, you know, if I were teaching my undergraduates, we would actually stop. We actually do every semester stop and have this conversation, but let's have it after I finish talking. But in your mind, what does it mean in your mind for research to have an impact? When does research matter? And you know, from Google, you know, some words come up. You know, be important, make a difference, be of consequence, be relevant. And even as a, you know, I am 100% scientist. I mean, I love my career. I challenge anyone else to come up and love their career more than I love my career. Like, I love science. But I still wonder constantly about whether what I'm doing is good enough, or is relevant enough, or has big enough impact. And I wanted to show you a sort of a counterpoint of two different moments in my career, which, interestingly, I did not tell Jimmy what I was going to do tonight, and he nicely set this up. And one was about a decade ago when I gave a TED Talk. And that's the picture up there on the left. And this was a talk by me when I was in my postdoctoral phase. And right at the instant of discovery, of being able to see one of the fastest movements ever described on the planet. So in that decade, about 1 million people have watched this little 10-minute thing that I did. And last year, I was in the limelight because a senator decided that my work is a waste of money. And so I did a little clip on PBS NewsHour. They have this sort of neat thing at the end of one of their shows every week where they ask someone from all different perspectives to just talk about something that matters to them. So I did a 3-minute clip on why what I do could matter and the sort of story of dealing with the politicians. And I went to Congress and, you know, I did a presentation and talked to these politicians. And in less than a year, over 3 million people have watched this little clip, which makes me wonder, you know, when we talk about what's important, is it about the work? You'll hear about it today. Is it about exciting someone's interest in a particular, you know, moment of discovery? Is it talking to politicians? Is it addressing this bigger issue of what science is about? And so I realized, you know, there are many ways to have an impact. And when we ask ourselves, what research matters, there are a lot of different answers to this. And I wanted to put up this quote that was posted under my TED Talk some time ago by someone who I do not know. Probably this will be recorded and he'll come out from somewhere. And he wrote, after watching the TED Talk, a bit worried that I'm finding this stuff interesting, although implication of that research and the benefit to mankind are probably close to zero. And one upvote note over there on the left. So this is a legitimate concern. If you're super interested in something, is that a problem? Right? So, or is that an impact in and of itself? That I made someone interested in some key part about biology. So this is the philosophical part of today's talk. And Jimmy had mentioned that there are folks at various points in their career in the audience, and I think this comes up a lot. In fact, a lot of people who emailed me about my PBS NewsHour were educators saying, wow, I wish more of my students understood that what is important is sometimes the knowledge itself. So today what I'll do is I'll talk about this arc of experience and while also telling you about my research. And while asking you to think to yourself right now what your thoughts are on this, and then I'll ask you again and then maybe we can discuss afterwards whether your thought patterns changed as I talked about today. So let's start with this field that I'm in. It's a field that studies how organisms move. So organismal movement involves figuring out the physics of how things how organisms are doing things. There's components that have to do with biomimetics and bioinspiration, which I'll explain in a moment. And there's even evolutionary thinking. And the cool thing about this realm is that there's a wide spectrum of basic and applied research, and a wide range of perceptions about what makes this research important or not important at all. And I wanted to start by just defining two things. So Jimmy used this word, basic research, and we use this word in our daily lives and in society, but it's not always clear that people know what it actually is. So basic research is defined as motivated by curiosity or interest-based scientific questions. It's justified by the goal of increasing human knowledge and understanding. It's not driven by a product, process, or commercial value, and it's the foundation for future applied research. In contrast, applied research is focused on solving existing human-centric problems, not justified by the pursuit of basic knowledge, often oriented toward human health, engineering design, and agriculture. What I'm going to show you today is in this wonderful world of science that I'm in, there is no clear distinction between basic and applied research. It's this incredible continuum, and that's a large part of what I want to talk with you about today. And finally, just another couple quick terms. One of the applied sides of what we do in this world of science, is what's called biomimetics and bioinspiration. And this is a very, very vibrant area in engineering right now, which is to look to biology for new ways of doing things or new design principles for developing better human products. So we'll even talk about that today, coming out of some of my work on mantis shrimp. And there are a few other terms that are slightly different there. So I'm going to actually start, I realize this is a marine lab, but I'm going to start with just a quick vignette about this continuum of discovery, basic research to applied research, and how it's played out in perhaps one of the classic questions, which is how animals fly. So we could start, right, by just looking at a bird, or even just looking at this photograph. I was looking at an osprey over dinner tonight, flying. You know, all that like characteristic wing flap that an osprey has, right? We can look at this owl and immediately ask, well, what are those like little wingtips doing? You know, those little things. Why are they spread? Well, look at these little foot warmers. Look at that. You know, the yellow eyes, this white striping. There's this bit of wonder, just immediately. That feeling of curiosity, of wonder. And I can tell you that a lot of the things that you and I are looking at right here, scientists don't know the answers to yet. Engineers would love to know all the tricks that this owl is playing to fly, right, quietly through the night. Take another totally separate, independent evolutionary origin of flight in bats. And here, you know, there are clues that connect us to this animal. Fingers, arms, skin. Dragging its feet through water, mouth open. The wonder of that, the beauty of that. Another origin of flight in this crazy grasshopper with beautiful rainbow-colored wings. Its antennae are up, blue eyes. So much information, so much wonder, so much joy in the knowing, in the wondering, in the possibilities of what this organism represents. What's neat is that there is a huge history of humans wondering about this, discovering about this, and having it end up being one of the most significant engineering feats. So Da Vinci in the 1480s was deeply inspired by birds in flight. He was attracted by their beauty, by their science, and the application. In fact, he wrote the Codex on the flight of birds. He was obsessed with kites and developed ideas for flying machines based on his observation of kites. Similarly, Otto Lilienthal studied storks. He thought that they might have the trick to how we could get humans off the ground and into the air. Our ability to fly the airplane that I was in today, was entirely inspired by human wonder of what these animals are doing that we can't do yet. Where does the basic research start and end and the applied start and end? I'm not sure that we can pull them apart. The Wright brothers were obsessed with banking turns by birds. They were sure that there was a trick in there that birds were doing that they could retain their control as they turned, as they worked on developing the flying machine. So even today, right, we've got things in the air because humans wanted to know how that could be possible. But check this out. Not long ago, we were at a point where the engineers understood the physics of how an airplane flew in the air, but if you applied those physics to a bumblebee, the bumblebee couldn't fly, right? So back we are again to the bumblebee asking how that could be possible. And so, of course, it was discovered, this is not going to be an aerodynamics talk, but it was discovered that insects are doing really tricky things with air, with the flow, with developing little vortices. Immediately, engineers want to know, can we use those tricky engineering tricks to get little things in the air with incredible control and efficiency? And there they are, right? New micro-flying machines. I had someone from the Department of Defense in my lab last weekend, and we were talking about how just look around you and look at what biology is doing, and we humans still haven't figured it out yet. That's why we need to be looking there, right? So again, all the way back, we get the machines in the air, our airplanes in the air, and then we come back and realize that the bumblebee still has more tricks than we can apply yet. So what is basic? What is applied research? Turns out the bumblebee trick, the things that engineers find incredibly helpful and illuminating for getting little things in the air, heck, seeds evolved to do as well. So even seeds are using aerodynamic tricks that we are only now starting to apply to flying machines. Seeds, of course, that want to, essentially need to stay in the air as long as possible to propagate. So this world of issues, of questions, of wonder, of applied, of basic to applied research are integrally connected in this field that Jimmy and I are in, this world of biomechanics, how organisms move. So discovery and application, basic and applied, iteratively incorporated. So I want to today show you how these all connect in, even if in the end, by the end of today, or you already knew for sure that what matters to you about research is that it has human applications. That is the right answer. Or if you are sure that research is here so that when you go out diving, and I'll show you later, actually momentarily, and you see a mantis shrimp and you want to know how it strikes, just because that is also the right answer, right? That's why we're doing this research, for all of these reasons. That's why scientists are doing this work. So, what I'll talk about now for the rest of this lecture is how I've integrated these issues, just the basic equations of physics, ever asking myself, is this a sufficient impact? Is this work that I'm doing good enough? Am I doing enough for the world? How we incorporate evolutionary biological thinking with design in a creative way. So I'll begin, like what I did, with the bird pictures and show you some pictures of mantis shrimp, because even though I actually work on a whole bunch of different organisms, tonight we're going to do mantis shrimp. If you want to talk about other ultra-fast organisms, I'll tell you about those afterwards. But here are some pictures of mantis shrimp. This one you might be familiar with. I was out snorkeling here last year, and they're very, very abundant, just up the road a bit. These are mantis shrimp that make burrows in the sand, and they look up at you, and you can barely find them. You have to get into a zen-like state and say to yourself, I am not seeing anything. And then they will all suddenly appear right in front of you. That's how tricky they are. These are mantis shrimp that live in burrows. This animal formed the burrow by hammering it. It's on a hard rock. This animal produces mucus to create these burrows deep in silty mud. And here's one of my favorites. This is actually a picture from California mantis shrimp. And this one actually decorates, it's in mud, and it decorates the outside. This one is not super decorated, but it pushes white shells around the perimeter. So it has a prettier entrance, which I always thought was cool. Again, with wonder, in fact, this whole line of research started with a simultaneous big failure and fascination. I was supposed to be figuring out how mantis shrimp produce sound and hear. Still haven't figured that one out entirely yet. While I was doing that, I realized perhaps I was watching the much more interesting question, which is how these animals were breaking open snail shells with tiny little hammers. Hammers that many of these hammers were on the order of the mass of 2 toothpicks, breaking open snail shells that I had to go and take my big hammer from Lowe's to break open in the water. It started there. All of this stuff that I've been working on for over a decade, obsessed, keeps me up at night, started with me just sitting there like, that is cool, right? So you'll see where it's taken me. These are pictures of the spearing mantis shrimp that are very abundant right here. These animals hunt from burrows and reach up and capture fish swimming overhead. Spectacular. So I, as a scientist, want to figure out how this is done. Other people get inspired to do artwork. I wanted to show you some of the things people have sent me over the years. This is an origami mantis shrimp. I think like a friend of my lab manager was complaining that she had to go in and feed, well, no, she wasn't complaining. This is a recorded lecture. I'll be very correct. She had an arduous weekend getting in to feed the mantis shrimp, and her friend made this drawing for her. Thank you for feeding us during the snowstorm, inspired by the beauty of these animals. A former undergrad made an entire set of cookies shaped like mantis shrimp hammers with the mantis shrimp in the middle, another drawing. So some people will take that and do artwork. I wanted to know how they were doing this. So this is the story of how it started with basic research and how we've iteratively moved back and forth, how I started at a TED Talk, ended talking to senators, and somewhere along the way, this research has made it to engineering, made it over to that side where new products are being developed with more on the horizon. So here's a picture of a video of a spearing mantis shrimp, like I just showed you, the ones that live in mud, the ones we have here, although this was actually filmed at night on the Great Barrier Reef. And you can see this animal hunting at night, catching stuff overhead. I dare you to put your GoPro out there, do a little filming at night. It's pretty spooky, but very fast appendages, catching little things going by. And these animals, as you probably know, if you don't know, you ought to know that you're swimming over out there, are a good 2 feet long, 1 to 2 feet long. They're huge. Yeah. When I was actually snorkeling out there working on them last year, there were all these kids and families around. I'm like, I don't think you know what's down there, or you wouldn't be here. So these are the ones that have the spears to capture fish going overhead. These animals, on the other hand, have a hammer on their elbow, essentially. And they break open snail shells. So this is an animal handling a snail shell that I would need to break open with a hammer, touches it with its antennules. Oh, didn't like the position, wiggles it into place, gives it a whack, and it just broke that snail shell in half. We can slow this down a little bit. This is a teeny tiny spear. It's an adult, only like a centimeter, 2 centimeters long. This one, you can see the motion a little bit better. We've slowed down the movement using high-speed imaging. And here's a smashing mantis shrimp knocking the claw off of a crab. Again, slowed down. These movements are invisible with your naked eye. So, just out of curiosity, I'm going to ask you, we've got mantis shrimp that can catch fish swimming overhead, and we have mantis shrimp that break open snail shells. Those of you who think the mantis shrimp that catch fish should be the fastest, raise your hand. Okay, it's not a trick question. It's a legitimate scientific question. How many of you think that the mantis shrimp breaking open the snail shell should be fastest? Okay, you all are way smarter than I am. Let's just put it that way. So back in the day, I worked on just figuring out how fast they were moving. And what I discovered is that the best smashing species goes at highway speeds, the acceleration of a bullet in a gun, in less than 2 milliseconds. This was one of the fastest described movements at the time. So I thought, well, heck, the fish-catching mantis shrimp must be much faster. So my graduate student, Maya DeVries, worked on this for years, and she kept coming back to me with these really boring numbers, 6 meters per second and the like. And finally, she came in and she plopped this whole data analysis from the literature, and she said, this was fast before you discovered ultra-fast. And she said, this is what we used to think of as fast. This is as fast as squid tentacles extending rapidly. This is as fast as a fish strike. This is really fast, isn't it, Jimmy? Yes. So Jimmy's animals, the fish over there, they're like this fast. What I'd realized at that moment was I had accidentally stumbled into a realm that we didn't really understand before, because our understanding in biology about fast was all about capturing prey that swim away or run away, or the cheetah, right? Fastest animal on the planet. Well, I am sorry to tell you, but the cheetah is not the fastest animal on the planet, not by a long shot. So I made this chart, and you don't need to read all this stuff, but what I realized is that we have a pretty wide range of durations. Nematocysts are the stinging cells of jellyfish. They are the fastest in terms of duration and acceleration by a landslide. So next time you're stung by a jellyfish, just celebrate that moment, okay? Speeds are all in the same order of magnitude. Cheetah down here, human runner. Cheetah and human runner are at the bottom of acceleration. Our squid strike is about a race car. Spearing mantis shrimp are around a missile. Smashing mantis shrimp are around bullets in the muzzle of a gun. Trap giants, which is a major focus of my lab at the moment, an order of magnitude higher, 2 orders of magnitude higher. So when I was having my conversation with one of the senators who felt that this work was a waste of money, and I pointed out that actually biology was doing this stuff way better than we could do, and not only that, but these animals don't self-destruct when they set off a gun, right? They do it over and over and over again for the lifespan. It started to click that, wow, biology might have really interesting insights. Now, right, you may be like, well, we don't need an even better gun, right? I'm with you on that. But the point is that physically, we have these capabilities in biology that we don't fully understand yet. So to boil it down a little bit more, what I realized is that we had stumbled on a world of different function than the way we had thought about speed before. Only down here are we looking at prey, capture, and jumping. Up here we have puncture. We have issues of Reynolds number for you physics buffs, impact, and launching. These animals and organisms are doing something different. So what are they doing? Back to our mantis shrimp. So here's a mantis shrimp. This one really does have a hammer, the mass of 2 toothpicks. I'm filming at 30,000 frames per second here. Actually, Suzanne, a former graduate student of mine, did this video. And what you'll see, this is way slowed down, is the hammer come in and pulverize this snail. You could jump on this snail shell all day and you would not break it. Okay, so this animal does this over and over and over again with no trouble at all. We can slow it down and it starts to reveal a secret. If you could see that flashing light, it turns out that not only are they hitting it at a high acceleration, but they're doing something funky with the water, and that is cavitation. So cavitation occurs when something moves through the water so fast that it creates an area of low pressure, and it creates a vapor bubble. It's kind of like putting a pot of water on your stove and heating it up and having it boil, except there's no heat applied here. It's because of extremely fast motion setting up low pressure. When that vapor bubble collapses, it emits heat equivalent to the surface of the sun, light, and sound. This is what, if you want to be a real tough guy on your boat and spin that propeller extra fast, this is what destroys boat propellers. It's that energetically intense. It is also why it is impossible to drive a submarine right now ultra fast through the water, because the water cavitates and it destroys the metal of the submarine, but it is also very, very, very loud. So being quiet in water and being fast is a serious problem, and it's an upper speed limit to even swimming fish as well as things like quiet submarines. We discovered this. And by the way, I discovered this because I was curious how they were breaking open these snail shells with a hammer. And I got some help from a BBC TV crew who happened to be willing to rent me the only video camera on the market at the time that could even visualize this motion because it was so fast. We see this very first video, and there it is. These animals are cavitating. They're literally imploding a bubble when they strike. So me being the tenacious 100% scientist that I am, I'm like, I want to know how much force this is. So that set me off on a whole other thing for quite a while, which is trying to measure the forces of something moving at the acceleration of a bullet that doesn't break it, that doesn't break the sensor, that can respond quickly enough. And this actually is another hilarious story that I would love to tell you, but I have a feeling I'm getting like super long-winded. But I'll tell you later if you ask me about it, how I happened to get my hands on this thing. So I'd broken a whole bunch of these things. I'm pretty much out of money, and someone rescued me on Friday night in New York City when I called the company. And I'll tell you the rest of the story later if you ask. So this is a special sensor, which is made of crystal, that is sensitive enough to detect a very high acceleration, very brief force. Also, just on the market when I started working on this. No one had been able to measure these forces before. You can also record the forces with sound because it's loud, right? So these animals, the technical part was really hard. The animals were happy to participate. So they come up and they hit it because they're mean, nasty, angry animals. So they hit it, immediately got data. We could also see that cavitation bubble is also forming on the surface of that force sensor. I was able to get my hands on an even better high-speed video camera, now filming at 100,000 frames per second. Your video, your phone is probably 60 frames per second. Maybe someone has one that goes 1,000, 100,000. At the time that I did this, nobody believed me, really nobody believed me, that there was a possibility that these animals were hitting with the appendage and generating force from a cavitation bubble collapse. So I had to actually show that this was happening. So this video shows it. Slowed down very, very much. There's the appendage coming in to hit the sensor. It hits, it bounces off, and we can see the cavitation bubbles forming and collapsing. Still not enough for my beloved skeptical colleagues. We love this as scientists. So I synced up the sensor and the video. And if you watch the top video, you'll see the force coming out. You'll see the force from the impact and the cavitation bubble, okay? So I gave you an average view, but oftentimes the cavitation bubble is 2.5 times the peak force. This is 800 newtons, and I'll show you what this really means in terms of the animal's body. So that's 2,500 times their body weight. Now, I got a 25-page handwritten letter from someone in prison. This actually happens to me reasonably often, I'm not sure why, who claimed that this was better than people who do karate or somewhere around there. Anyway, I would love to have that validated by someone who's in the free population. So apparently this is really impressive, okay, in terms of how many times body weight. So peak impact forces, they have two hammers, right? They have two appendages. So one snail shell hit gets you impact, cavitation, impact, cavitation. Let's compare this to what we knew about animal forces in terms of breaking open snail shells before I did this work. And that is animals that eat, other animals that eat shells. And a typical one is a horn shark. They're pretty great. Are there horn sharks here? I don't know. Anyone know? They're really cute. They're like this big, and they eat snails. So one of my colleagues naturally put a force sensor in the mouth of a shark. That's what we do, right? So if you look at this, though, this horn shark is biting, so the force goes up as it bites, and the force comes back down as it lets go. This is in milliseconds. This is in microseconds. The entire mantis shrimp strike happens here. I happen to pick a sort of pathetic mantis shrimp strike here, around 200 newtons. You just saw one that was 800, to match it up. This is a 3-kilogram animal. This is a 40-gram animal. Okay, so what I realized is that these animals had evolved a totally different strategy for getting into a snail. A snail that could be only opened by a much larger animal. I'll show you that in a slightly different way. So if we look at the massive animals that chew on snails, I kid you not, all of these data points are from my fellow scientists who put force sensors in the mouths of snail-eating animals. I think that is so cool. This is their peak bite force. Up here are things like alligators. If you go across, it lines up with a mantis shrimp. A teeny tiny mantis shrimp can generate peak forces of much, much larger animals. So this is an alternative strategy, extremely lightweight, very low energy. For those of you who have a physics bent, you notice underneath those curves, the area under the curve for the mantis shrimp is very small, very low impulse, but very high peaks. You know, this is very cool from an ecology point of view, but my colleagues who work on ballistics, who work on body armor, who work on traumatic brain injury, that interestingly involves impacts and cavitation in the brain, struggle as well. Still, we do not fully understand how these tiny, relatively tiny energy, but high peak forces can generate such an incredibly catastrophic failure in materials. So this is a very rich area, and I really do. I have literally collaborations, ongoing collaborations with an engineering group that builds bulletproof vests, and another group that works on traumatic brain injury, because we're actually dealing with the same physical problems that people have not fully figured out yet. But let's come back to this realm that I started with, which is, here are the mantis shrimp, okay? They're up there doing bullet-like stuff. Now, many of you are old enough to know that you do not get your cake and eat it too in life, right? You get one thing and you lose something. This is definitely true for me. Maybe someone out there has not discovered this yet. But there's that feeling. Well, this is also true in physics. It's also true in biology. Because an obvious thing is to say, well, why don't mantis shrimp take over the planet? If you actually think this might be true, you would be right in line with a science fiction writer who has written 2 books about mantis shrimp taking over the planet. It is worth a read. I love it. The final character is this like 6-foot-tall mantis shrimp with shimmering rainbow hair. I love it. I bought copies for everybody in my lab. I have multiple copies at home. But the thing is, they haven't taken over the planet in reality. Not in the science fiction book, they were doing a good job of it. And it's because you lose something when you get something. So let's take a look at a few of the costs. Why isn't everything doing this? What are the problems? And let's start with one that mantis shrimp have in a way solved, and in a way something that I think humans can connect to quite well. So mantis shrimp have to get along. They have to get along to continue the reproduction, right? They have to settle disputes about territories. They have to live next to each other. But they have lethal weapons. One blow by this animal on this animal would kill it, right? And I've seen it. I've seen them do this. One blow, and their conspecific or whoever is dead. So over time, these animals have evolved a way to not kill each other and still fight. So the way they do this is they have a special piece of armor at the end of their tail, and they hit each other back and forth on this piece of armor until somebody wins. And what we discovered through the work of my graduate student, Patrick Green, is that they will typically encounter each other, they're visually signal, and then they'll perform this ritualized fighting. So this animal is on the receiving end, this one's coming up with these bullet-like accelerations, and they fight back and forth. And what he found is that they essentially spar. For those of you who are interested in that kind of sport, this means that these animals are winning by the number of strikes, not by how much force capacity they actually have. So much like classic sword fighting and so forth, generally speaking, the sport today is try to get as many hits as you can, but not to actually gut your opponent. Same thing here. So the number of strikes, not the strike force, predicts the winner. This is kind of neat. But, when we take a high-speed video of this interaction, these animals are hitting this piece of this piece of armor with cavitation, with bullet-like accelerations, and it's not breaking. So now we have another question, which is, why is this not breaking? Because they can break a snail shell. So all of a sudden, we have the potential for a new idea about armor. So we've studied it in a somewhat preliminary way, led by a former postdoc of mine, Jennifer Taylor. She's now a professor at Scripps. And we discovered that not only is it a really neat piece of armor, and in fact, the impact mechanics is very much like a baseball hitting an ash bat. This is a little bit hard to come up with analogies for, but on one metric, this is essentially what it's like. So it's bouncing off, but there's a lot of energy that goes back into the bat being this part. But here's the crazy thing. We did these tests all along the animal. And out here on its mushy parts, the back and its abdomen, the impact has doesn't change with the size of the animal. But the impact against the armor scales with the size of the animal. So not only does the armor resist this impact, it actually contains information about the body size of the opponent. So, maybe some lessons for us here on how to continue to resolve our conflicts in human society, but also maybe some clues for armor, smart armor. Okay, now to some costs. These animals move so fast, the animals that I study move so fast that they're blind to the motion. Our nerve signals that travel through our body are slow by comparison. So slow that we can't see these movements, but these animals can't even sense them. It's done before the neural signal is down there and has a chance to get back to the brain. So what this means is that these animals are essentially doing something like a bow and an arrow situation, or even a bullet, where you have to actually load everything up ahead of time, aim it, and let it go. In the case of the mantis shrimp, the thing's still attached, but they're brain-wise blind to what's happening. So all of the planning has to happen ahead of time if they're going to do something clever with how they're processing the prey. And indeed, that's what they do. So this is the work of a former postdoc of mine, Katsu Kagaya. And he did a bunch of work looking at the activity of the muscles. These animals use a spring and a latch system, very similar to a bow and an arrow. So muscles are totally unable to actually generate this motion, just like if you try to throw an arrow at a deer, you're not going to take the deer down, but you can use that exact same muscle, load up a spring, and release it with the latch of your fingers and have that arrow move at those speeds. So essentially, a lot of my work is studying the bow and arrow latch versions in animals. That's what these animals do. So it turns out that they can adjust the spring loading in their appendage and release it to get variable outputs. Okay, now that's great news. Got us a good paper. But then the question is, how do they know what the heck they did? Right? So how smart can a mantis shrimp be? Right? Like now they're looking at a broken snail shell. Do they know what they did? Could they learn from it? Why are they adjusting it to begin with? The answer is I don't know to any of those questions. So any of you want to come and help me with that? Come on by. Just got to get a little bit of funding first. Okay. Cycling back here to the cavitation thing. Now, I want it to be magical, right? That mantis shrimp use cavitation to break open hard-shelled prey. But the truth is, we know that anything moving fast enough through water will cavitate, whether it's a submarine, a stick, boat propeller, whatever. So a former graduate student of mine, Suzanne Cox, an absolutely brilliant machinist and scientist and engineer. She was actually trained, she had her undergraduate degree in physics, her master's degree in engineering, and then decided to get her PhD in biology. And now she's working on really cool stuff with external efficient prosthetic devices for helping humans walk more efficiently, but also in general for people who need prosthetics. Now, so she's basically working on the interface of engineering and human health. Amazing person. So she decided, though, that she wanted to know whether mantis shrimp were intentionally generating these cavitation bubbles or whether it was an accident. So she built NinjaBot. NinjaBot is essentially our mechanical version of a mantis shrimp in order to control the system a little bit better. And that means that this little device here is moving at the acceleration of a bullet. And we can swap out mantis shrimp appendages or whatever materials we want to study the impact. It took us 9 kilograms of steel to build what a mantis shrimp does in 0.4 grams. Before we built this, we were told actually by multiple engineers that this was impossible. And I went back to the engineers and I said, I am a good scientist, I am not an engineer, but I know that it's possible because I measured it in mantis shrimp. So she built it, but here we are, limited by our materials. We couldn't answer, we haven't, I can't say, I will never say can't, but we have not answered the question of whether the cavitation bubble is controllable or not. But we stumbled on something else entirely, which might even be more relevant. Which is, it turns out that when mantis shrimp are rotating towards the prey, they ought to be cavitating, just like that boat propeller does, but they don't. So mantis shrimp are doing something very, very tricky about their path through the water so that they can move so fast without generating cavitation, which would be very damaging to the rest of their appendage, and then can only having cavitation happen when it's useful to them. So my dream is to figure out what they're doing here dynamically and then help design a better boat propeller. A quiet one, a faster one. We'll see. I want to end with something kind of fun and then loop back around to where we started the talk today. My former lab manager, Rachel Crane, she's now a graduate student at Stanford. She came in, and my lab is populated by a whole range of random people who I dearly adore. So I've got neuroscientist, engineer, physicist, paleontologist, biologist. Rachel came in with a psychology background. And she came in and she's like, you know, these animals must know something about these snails. They must have some kind of mechanical intelligence. So she decided to figure out whether they know what they're doing when they try to break open a snail shell. So she filmed them, and you'll see, this is one of the videos she made. So this animal's just hit a snail. It's a nice conical snail here. It's working it, spinning it, getting it in place. You know, and they take a long time with this. I decided not to worry. Oh, it cleaned its eyeball. Important to do right in the middle of doing this, right? This is a short version, but sometimes they take a long time trying to get it set up. Oh, little hit, no luck, fell over a little bit. Wiggle, 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 touch, touch, touch. Notice that they actually are not even looking at the shell for the most part. They're touching with their antennules, and it just broke off the top. And look at that happy mantis shrimp. It's eating now. It's like, yum, right? Happy mantis shrimp. So what are they doing? It turns out they do have an algorithm, and an algorithm that depends on the shape of the snail. So they adjust what they're doing depending on whether it's a high-spired snail or a globular snail. And we're just at the final stage of writing this up for publication. I'm really excited about it. So they have a way to do this. What we want to do is to go, my engineering colleagues who work on impact mechanics of fracture, who don't fully understand how that works, can't tell me where you should hit a snail, or where you should hit anything, frankly. And what we're trying to do is get the animal to tell us what the strategy is for breaking open ceramics, so that maybe we can get the physics and engineering to meet us halfway, so we can understand what's happening in between. Okay, I love my lab. I love the people in my lab. if this wasn't enough, and finally, this is my final goofy thing, because it is an evening seminar, and it's actually not that goofy. Samantha, future pediatrician, okay, she's graduating from Duke. She wrote her senior thesis on this. She decided, we can do something even more clever with those mantis shrimp than give them snail shells. Let's use the Patek Lab 3D printer. So she made models. And these animals are just so ridiculous that they're willing to do it. So there's a cylinder, and it's got a hole in the side full of food. That animal knows perfectly well that the hole in the side has food in it, but it knows something about breaking stuff. So instead it finds the edges and tries to break it open from the side. So what we realize is we can actually use 3D printing to get into the mind of the mantis shrimp, to figure out what their algorithm is, so that they can get it open. I heard someone say, that must be frustrating. Yeah. But they just keep at it. Isn't that great? So she's just finishing up her senior thesis on this now. We have really nice results. The short version is that smashers know to hit edges rather than the hole containing the food. And that's quite something for an animal, to avoid where they can feel and taste the food, knowing that if they work the perimeter, they could potentially get what they really want. Okay. So I started here. I started the talk here. This moment early in my career, it was an incredible opportunity to give a TED Talk where I was just in that moment of discovery, okay? And then more recently, realizing that there's a national conversation ongoing, and has been for a long time, about why discovery may or may not be without implication. Well, here's a crazy thing. So I gave this TED Talk, and an engineer who I'd never met, I still haven't met actually, saw this TED Talk. So Jimmy was saying that he spent maybe not all night watching TED Talks, right? But there are a lot of people, scientists and engineers, who watch other science and engineers talk to the public, right? And learn about science and engineering that way. Like, we're big consumers of this stuff. He watches my talk about mantis shrimp breaking open snail shells. And he did a podcast on this or something, and so I heard him talking about it. And he's like, wait a minute. Engineers said that snails are the most unbreakable material. So engineers are constantly studying snail shells. And he's like, I just watched a TED Talk with this lady talking about a shrimp that breaks snail shells. Now, I had no idea at the time that engineers only cared about snail shells. But by making this discovery and communicating it outward to the world, someone who knew the problem much better than I did looked in and said, we should be studying the hammer of the mantis shrimp, not the snails. And he has. And in fact, they have now made new materials, new lightweight, impact, fracture-resistant materials that they're hoping to put into things like football helmets and gear. So when we talk about what is the impact of the research, which of this is basic, which of this is applied? And I think the answer is I don't know. Did I know at that time that any of this had any implication, as this person pointed out, to the benefit of humankind? I certainly didn't bring it up, as he's pointed out quite effectively here. But in the end, it has. It's become really interesting. So my answer to the question, because I do ask it myself a lot, why is this stuff important? Why is it important to me? A huge part of it, of course, are all the people that I showed you along the way. We work with high school students, undergraduates, high school teachers for 5 years now. I've had a high school teacher in the lab. In fact, one of my high school teachers did a project in the lab. He was a physics teacher. He said he'd never done a biology class, never talked about biology in his physics class, and that's why he wanted to come to my lab. So he developed a curriculum, published a paper, I kid you not, on the day that I was up in the Senate office building meeting with members of Congress about this issue. He was over at the White House being recognized with his high school students for his contributions to STEM and encouraging young people to come through and become scientists. It was one of the most bizarre moments. Here I am trying to keep myself together, talking to these politicians about why my work actually is worth funding, while he's texting me talking about, looking through the White House and the people who were there and all these young people who are being encouraged to become scientists. So for the young people here and the not young people here, I guess my answer is, any answer is good, whether you want to make a new material or pursue that curious thing like I did a decade ago. So to end, my answer, is training, bringing people through, giving people the experience of science in my lab, but also to understand the Earth's phenomenally long biological history, to enrich our daily experience and our present biological world. I love that this is us in the tree of life. I love that we live on a blue planet and know very little about it yet. I love that mantis shrimp are so diverse. We've got spears, hatchets, hammers, so I could do the deep dive into the weirdness of the little critters that I study. I also think that we have the potential to discover new model systems with the power to inform medical research and improve the quality of life. I think this is also important. All of these major model systems that biologists use today that are dealing with and providing answers to us for our major medical issues started with a scientist being like, check that out, zebrafish are kind of see-through. Go check out Jimmy's lab, he works on zebrafish. Major model system. And finally, making use of the 200 million years of engineering experiments already performed by biology, bio-inspired design and biomimicry. One of the top award-winning new materials was designed from gecko feet, and an understanding of physics and how the systems evolved. And now in the mantis shrimp, we have new materials being designed by engineers, new possibilities for boat propellers, weird things about armor. We've got fossils of mantis shrimp, trying to figure out what was going on 50 million years ago when the hammers evolved. This, I think, is what science is and why it's important. So hopefully that was a bit of philosophy, a teeny bit of politics, not too much, I'm sure we're all sick of that, but hopefully all of you from your different perspectives on science could see some of the relevance there. And there are many funding agencies and people to thank, and I'll take the final few minutes for questions, and I'm also happy to hang out and just talk. I got a rental car, Jimmy can go home and go to sleep, watch more TED Talks. We can just talk. I'd love to hear what your thoughts are. Thank you.